Hello and welcome to today's practical on measuring the rate of photosynthesis. So, get ready for an exciting and hands-on exploration into the fascinating world of photosynthesis. So far, we've looked at the core concepts surrounding photosynthesis, which is a vital process that powers life on Earth by converting light energy into chemical energy. We've explored how plants, like the ones we'll be studying today, produce oxygen as a byproduct of this process. Specifically, we've discussed the key factors that influence the rate of photosynthesis, such as light intensity, temperature, and carbon dioxide concentration. Today, we are going to build on this foundational knowledge by conducting a practical investigation. By manipulating the light intensity, we'll observe and measure how it impacts the rate of photosynthesis in an aquatic plant, which brings the theoretical aspects we've covered into a real-world context, giving you a deeper understanding of how photosynthesis rates can be quantified and influenced by external factors. So, let's dive in and begin our quest to uncover the intricate relationship between light intensity and photosynthesis. Ready your notepads, microscopes, and measuring tools. Let the experiment begin. Before outlining the practical, let's take a moment to consider its importance. This practical experiment allows you to directly observe the process of photosynthesis, which is fundamental to all life on Earth. By investigating how light intensity affects the rate of photosynthesis in plants, you are studying the same processes that sustain global ecosystems. This knowledge is crucial for understanding broader environmental issues such as climate change, agricultural productivity, and ecosystem health. Grasping the mechanics of photosynthesis can help you understand how plants contribute to the oxygen supply and help take up carbon dioxide, maintaining the balance of gases in our atmosphere. Conducting this experiment sharpens your abilities in designing and conducting scientific investigations. By manipulating the light intensity and observing changes in the rate of photosynthesis, you learn how to frame a research question, control variables, collect data, and interpret results. These skills are foundational not only for future scientific studies, but also for any field that requires critical thinking and methodical problem solving. This practical offers an invaluable opportunity to apply theoretical concepts from your biology curriculum in a hands-on setting. By observing the production of oxygen bubbles in an aquatic plant, the abstract process of photosynthesis becomes concrete and easier to comprehend. This integration of theory with practice bridges the gap between what you learn in textbooks and real-world biological processes. Let's dive in and explore how light intensity impacts the rate of photosynthesis in plants. Let's set up our experiment to investigate how light intensity affects the rate of photosynthesis in an aquatic plant. Carefully follow the steps to ensure we achieve accurate and reliable results. Sam will go through the steps with you. Over to you, Sam.
So for this practical, first we set up the plant. Start by placing an aquatic plant, such as pondweed, into a test tube filled with a 1% sodium hydrogen carbonate solution. This solution provides a constant supply of CO2, which is crucial for photosynthesis. Now we will look at how to position the lamp. The lamp should be positioned at a specified distance from the test tube. Ensure you measure this distance precisely using a ruler, as it will be our independent variable.
Once everything is in place, turn on the lamp. Observe the plant for a set period, for example, 1 minute, and count the number of oxygen bubbles produced. These bubbles are a direct indicator of the photosynthesis rate. To ensure accuracy, repeat the bubble count 5 more times. Calculate the average number of bubbles produced per minute. Calculating an average rate of photosynthesis will make your experiment more reliable. Now we will change the distance of the lamp from the test tube to vary light intensity. This could involve moving the lamp closer or farther away in set increments and repeating the bubble counting process each time.
We can also use an alternative method to measure the rate of photosynthesis using a syringe. For a more precise measurement, connect the test tube to a syringe. As oxygen bubbles are produced, they will collect in the syringe, allowing you to measure the volume of oxygen produced over a set time. Now, throughout the practical, ensure that other variables besides the independent variable, light intensity, are controlled. First, ensure to use a water bath to maintain a constant temperature, as temperature can affect the rate of photosynthesis. Additionally, use the same concentration of sodium hydrogen carbonate for all trials to ensure consistent carbon dioxide availability. Lastly, use the same type and size of pondweed throughout all experiments to keep biological factors consistent. To analyze the results, follow these steps. Firstly, record the average number of bubbles. For each distance of the lamp, which correlates with different light intensities, note down the average number of bubbles produced per minute. Secondly, calculate the rate of photosynthesis. Using the number of bubbles and the time taken, calculate the rate of photosynthesis. The rate of photosynthesis equals to the number of bubbles divided by time. If instead you are finding the volume of oxygen produced using a gas syringe, you can find the rate of photosynthesis using volume of oxygen produced over time. Next, compare rates across different light intensities. To do this, create a table with columns for distance from lamp, average number of bubbles per minute, and calculated rate of photosynthesis. Now we will represent the results graphically. Plot a graph with light intensity or distance from the lamp on the x-axis and the rate of photosynthesis on the y-axis. This helps visualize the relationship between light intensity and the rate of photosynthesis. Next, we do the discussion. Let's discuss how light intensity affects the rate of photosynthesis. Typically, an increase in light intensity will increase the rate up to a certain point, after which other factors may become limiting. At this point, moving the lamp closer to the plant is unlikely to affect the rate of photosynthesis. Finally, consider any anomalies. If some light intensity levels show unusual rates of oxygen production, consider possible experimental errors or other external factors, such as inconsistencies in light exposure or variations in plant health.
In today's lesson, we conducted a practical experiment to investigate how light intensity affects the rate of photosynthesis in an aquatic plant. The key steps of this practical were: setting up. We prepared the apparatus by placing an aquatic plant in a beaker filled with water and added a sodium bicarbonate solution to ensure ample carbon dioxide supply. Varying light. Intensity. We positioned a light source at different distances from the plant to change the light intensity and monitored the setup each time. Measuring oxygen production. We measured the rate of photosynthesis by counting the number of oxygen bubbles produced by the plant over a set period of time. Recording data. We recorded the number of bubbles produced at each light intensity to analyze the effect of light intensity on the rate of photosynthesis. The topics this practical covers includes the process of photosynthesis and the role of light in this process, how environmental factors, such as light intensity, can impact the rate of photosynthesis. Our key takeaways for future practicals are to carefully control other variables such as temperature, carbon dioxide concentration, and type of plant to ensure reliable results. Accurately measure and record the rate of oxygen production to determine the rate of photosynthesis. Understand the relationship between light intensity and photosynthesis, which can aid in optimizing conditions for plant growth. This practical not only reinforced our understanding of the photosynthetic process and the critical role of light, but also emphasized the importance of controlled experimental conditions to derive meaningful scientific conclusions.